Thank you. Okay, so we know that we live in a very rich and complex auditory environment, but in that rich environment, the sounds that are in there are not processed equally. And there are many factors that influence which sounds are processed better and which ones are processed worse. We've heard already something on statistical irregularities, of course pop-outs on sound intensity, for example. But one factor that I would like to focus on today and in my talk is that of feature selective attention. So with feature selective attention, we can actually flexibly prioritize specific dimensions of a sound. So for example, we can prioritize processing of specific locations, sound frequencies, modulation rates. And that allows us to very dynamically and rapidly optimize the processing of sound input that is at that moment relevant. As such, it really it's a skill or an ability that helps us to process sounds and noise. So we've heard a lot about hearing and noise already over the past few days, and feature selective attention can really help to basically enhance or boost a signal that is currently relevant and suppress the currently irrelevant signal. But it's important to point out that feature selective attention can also work when there's only one stream, sound stream present. So it can also work when there's no noise in the background by simply kind of tuning into a relevant feature and responding more quickly or more accurately when a sound with that feature pops up. Feature selective attention has been studied widely, first in the visual system, but then also in the auditory system. And there are many people in the room today that have worked on it in one way or another. And I'm sure that I'm going to forget many or I basically don't have time to mention many of the relevant work that has been done. But I wanted to focus on work of kind of a single group of people that have been published over the last 20 years or so, coming from the lab of Shiab Sharma, Jonathan Fritz, Stephan David, who have shown ample evidence of rapid changes in the auditory cortex of the ferret with changes in task demands. So they have shown, amongst others, that there are rapid changes in frequency preference and selectivity of auditory cortical neurons as task demands change. They've shown that these changes are already present in primary auditory cortex, but that they are stronger as you move up the auditory hierarchy. And interestingly, that the exact changes that you do see in receptive fields and responses depend very much on the exact task and on the reward structure. So this is work from Stephan David, showing here SGRFs under different conditions. In the first column, passive listening, the second one, an active task, and then the third column, the difference. And the important modulation here is that the task and the reward system differs. So an approach task versus an avoidance task, where in the approach tasks, animals were rewarded when they licked, when they performed behavior, when they detected a target sound. While in the avoidance task, they were actually punished if they did not refrain from licking when they detected that target. And interestingly, both cases had a specific frequency band that was relevant here, indicated with the red arrow. And you see that in both cases, the SGRFs change at the frequency that is currently relevant, but in the opposite direction. So you see suppression in the approach task and actually enhancement in the avoidance task. Now, these frequency selective attention and in general feature selective attention has also been studied extensively in the human using non-invasive imaging tools. So mentioning some studies here. From human neuroimaging, functional MRI specifically, actually a very uniform picture emerges, and that is that of enhanced responses to currently relevant sound frequencies. And across studies, also the presence of receptive field changes or populations of receptive field changes in the case of functional MRI have been studied, but there has been no evidence for such changes being present. So that actually means that there is a bit of a gap between these two lines of research, where from the animal electrophysiological side, we really see very dynamic, very diverse changes also, while the human neuroimaging results really show this uniform picture of simply stronger responses to attended sounds. So we set out to try and kind of fill this gap and see if we could contribute to this apparent difference. We had setting out a number of reasons in mind why these differences may exist, because there are simply very large differences between these between these research lines. First of all, of course, the methodology, the spatial resolution of invasive electrophysiology is very different from that from functional MRI. As was already mentioned earlier today, we have a voxel with functional MRI with thousands of neurons in there, so that could definitely mask a lot of changes that actually can be seen with invasive electrophysiology. A second potential difference is that of spectral resolution. So especially if we would look at frequency, changes in frequency preference, changes in frequency selectivity, we would like to actually be able to map our responses in a voxel, in this case, at a very high spectral resolution. But for those of you that have worked with functional MRI, you're very familiar with this likely. Any condition that you add, and in this case, any frequency band that you add that you want to map, that's going to take time, right? So functional fMRI is sluggish as it is, so you're collecting data already rather slow. You'll have to repeat it a number of trials before you get any reliable signal to begin with. So that means that to keep things feasible for a subject, generally the spectral resolution with which you could possibly map changes in voxel tuning is low. And then finally, there's a difference in task design as well. So looking a bit more in depth into what these studies actually did, and the studies I talked about coming from an animal electrophysiology, the sounds are actually presented sequentially. So there's only a single stream of sounds where an animal tunes into a specific sound frequency, for example, and responds whenever they actually hear that sound coming up. Instead, from the human neuroimaging literature, there are always at least two streams present. So there are multiple streams present, where in one of these streams a relevant target appears, and the other stream it does not, or a target should be ignored. Okay, so what we did was to try and set up a functional MRI study that, in terms of stimuli and experimental design, would move as closely to the animal literature as possible. And we come up with the following. Participants listened to artificial ripples and natural sounds, but in a single stream. So there was only one sound present at a time, and over time this varied. So I'll let you listen to an example.
Probably the usual.
And this went on for a very long time, okay? And they had to perform a noise burst detection task. So within the ripple sounds, but not the natural sounds, within the ripples we would embed very short noise bursts, 112 milliseconds of duration, a white noise, and we varied them in intensity and basically made sure that at individual levels, participants could detect them accurately in 60% of the trials. Then these ripple sounds, they came in two flavors. They were always an octave broad, but they were centered either on a low frequency, 300 hertz, or on a high frequency, 4 kilohertz. And over the course of the experiment, the noise bursts were either more likely to occur in the low frequency ripples or the high frequency ripples. And we first checked behaviorally if this manipulation indeed worked. So if behaviorally we could see an advantage of participants attending the low or the high frequency ripples. And that was indeed the case. So what you can see here is on the y-axis reaction time for noise bursts embedded in low ripples or high ripples. And you can see that indeed participants responded faster here in the gray bars to noise bursts and low ripples in the attend low case, and faster to noise bursts in the high ripples in the attend high case. And we could also replicate this behavior in the scanner. In the scanner, though, we had to present these noise bursts at a slightly higher intensity, so we had to go to 80% detection threshold, because otherwise participants could no longer do the task in the scanner. But other than that, the behavior was fine. We then started to look at the recorded functional MRI responses, and we had a big surprise. So this is the difference between responses to ripples when they're attended versus non-attended. And based on all what we had seen previously in functional MRI studies, we had expected a stronger response to attended ripples, but we saw the exact opposite. So we saw a weaker response to attended ripples. Now, we did a number of checks to make sure that this was actually the case, and I can tell you more about that if you would like to know, but we checked over and over, but these responses were indeed lower with attention. So then, in order to figure out why this could possibly be the case, we turned to the responses to the natural sounds that we had recorded. And we did population receptive field mapping. So Ione already did a great job in setting me up for this, but we did a slightly different thing to what her lab normally does, because we did population receptive field mapping not on artificial sounds, but on natural sounds. Now you can nicely do this as well, because natural sounds, of course, we know what frequency content they have, and each sound has a slightly different frequency content. So what you can then do is basically create your set of hypothesized tuning curves for the voxels. So these are one-dimensional Gaussians that have a mu, a mean, that would be the preferred frequency of a voxel, and a sigma, a tuning width of a voxel. And then you basically just pass your sound spectra to a hypothesized receptive field, and you will get a predicted response. Now, if you do this for your whole library of basically predicted receptive fields, right? So you can, we ranged from best frequencies from about just under 200 hertz to 8 kilohertz and a width of 0.3 approximately to 3 octaves, then you can get a lot of predicted responses, right? So this would be a different best frequency, and this would be a difference in tuning width. You get a lot of predicted sound source responses to all of your natural sounds, and then you do the same thing. You get for each voxel an actually measured sound, and you correlate that measured response to all of your possible predicted responses, and that will give you a correlation matrix with the best frequency mu and the tuning width on the and the tuning width sigma on the y and x axis, and the kind of hotspot in that correlation matrix will tell you the mu, the best frequency, or the estimated best frequency of the voxel, and the sigma, the tuning width of that voxel. Now, importantly, what we do is that all of this procedure is done on a subset of our data, our training data, and then we take, for each voxel, their estimated mu and sigma, and we see how well we can predict using that mu and sigma responses to sounds that were not used in estimating them. So this is what you can see over here. Now you see the correlation that we observe in each of our participants between the predicted and observed responses to testing sounds given the mu and sigma of each voxel in training. There are two things that are important here. First of all, we're above 10, so that's good. But also, we are relatively stable across participants, and the most important thing is that there is no difference between the two relevant conditions. So across attend low and attend high, we're actually performing equally well in PRF fitting. Okay, so what can we do with this? First of all, we can make a tonotopy and tuning width map, so that's always nice to see. This is a single participant, and a few things to observe here is that the patterns look as they should look. So we have, a low frequency tuning on Hassel's gyrus bordered anteriorly and posteriorly by tuning to higher frequencies, and a tuning width that shows a narrower tuning on Hassel's gyrus, and again anteriorly and posteriorly, laterally broader tuning. Also, what is nice to see and interesting, I think, as well, is that actually these are three different sets of data for the same participant, where we had a baseline data. These are two separate sessions of sounds where only natural sounds were played, so no ripples. And then here the attend low and the attend high, which are basically separate parts of our data. And you see a lot of consistency in these maps. You do see differences as well. And then, of course, the big question is what of that is simply noise and what is actually an interesting difference. So we returned then to our unexpected finding, and what we could then do after this PRF mapping is that we could actually look at these voxels that showed a difference in response to ripples when they were attended versus non-attended, and check what kind of characteristics they had. And what we saw was that the difference between attended and non-attended responses to ripples, and specifically this response suppression of ripples when they were attended, was mostly present in voxels that had a best frequency close to the target frequency. So the closer that voxels would get to the target frequency, and that, wrong button. And that was the case both for the 300 hertz and the 400 hertz ripples, so respective to voxels that had a best frequency of 300 hertz and 4 kilohertz. The closer they were to the best frequency was to the attended target, the higher that suppression was. We observed a few other things. So we saw no effect on gain of receptive fields, voxel receptive fields, no effect on best frequency either. So we did not see shifts in best frequency of voxels across attention. We only saw this narrower tuning width in locations whose best frequency closely matched the attended frequency. So what we think is happening is that the narrowing of tuning width in these voxels with attention actually causes or results in this lower response that we observe to ripple sounds. Now, what does this mean? Our results match, or at least are not contradicting the findings from animal electrophysiology, but they do actually very much contrast with those of previous functional MRI studies. We interpret that, or we can interpret that, by going back to our original hypotheses that the difference between actually originally animal electrophysiology and functional MRI may have come from one of three factors: spatial resolution, spectral resolution, or difference in task design. And what our results show is that we can pretty much exclude, at least from our perspective, these first two factors, because we are now seeing in our data a difference with functional MRI studies, even though we are working with fMRI, so the spatial resolution should fall out. The spectral resolution and the spatial resolution also can be excluded because of the fact that we're actually seeing a very big difference, not just in the receptive fields, but actually in the overall response to sounds, which is lower in our case with attention compared to higher with attention as in previous studies. What we believe we're seeing is actually a result from a difference in task design that can explain both the difference of our results to previous fMRI study, but also the gap that was there between the two fields. So what we think and what we'd like to propose is actually happening is that feature selective attention and frequency selective attention in this case can work in different strategies depending on what is useful at that point. If there are multiple streams present, frequency selective attention may work as a competitive enhancement. So the sounds that are there compete for limited neuronal resources, and a boost of the attended stream may be most useful to actually give the sound that you're currently attending to, the sound that is currently relevant, the boost that it needs to compete to the other. However, when you're in a single stream condition, it may be very different because you may not need that boost. Actually, the most important thing may be for you to sharpen tuning to the relevant frequencies and suppress off-target frequency ranges to reduce the chance for false alarms. Okay. So in the minutes that remain, I would like to switch gears a little bit and ask a different question. Where do these changes come from? And of course, we know that in the end, if we are talking about top-down selective effects, they must, and attention, they must come somehow originally from some top-down projection. But what I actually mean with that is what is the neural circuit that is driving these changes in receptive fields and population receptive fields that we're seeing? And for that, and this topic was already nicely set up by Jean-Marc Adeline, who talked about the amount of feedback that is actually there from cortex down to earlier regions. And inspired very much, again, by animal electrophysiology, the work of Dan Pauly, also the talks that we heard earlier, the speaker Ross Williamson, work of Vittorio Bajo, made me very interested in a specific circuitry. And that's the one that runs from deep primary auditory cortex, so these are layer 6 corticothalamic cells, down to the ventral part of the MGB. And interestingly, these projections are tonotopic, and they actually come, so if you have a neural population here that projects up to middle layers of primary auditory cortex, then the layer 6 neurons that come down, they actually project back to pretty much the same location. So it's a tonotopic projection. That's an excitatory feedback, but there is another feedback signal coming in because these projections, they actually branch through the thalamic reticular nucleus. That's a thin shell that sits all around the thalamus, and it has sectors for different senses, including an auditory sector, and that actually projects in an inhibitory fashion back to the ventral MGB as well. It's also a tonotopic projection, but a much broader tonotopic projection. And this circuitry, as was proposed very nicely in a recent review, may actually be perfect for setting up any type of population, well, receptor field changes, or in our case, population receptor field changes that we could see with feature selective attention. So by strengthening the feedback from primary auditory cortex to ventral MGB, you may get a gain, so a boost in signals as needed, as frequency selective signals as needed, but actually by instead strengthening the projections back from TRN that are inhibitory to ventral MGB, we may get narrowing or even shifts in frequency preference. So what I hope to do in the next few years, and what I would like to show you the initial results of next, is to actually see if this system can underlie the changes that we see with attention. It's not an easy thing because these structures are very small. But we think that with ultra-high field functional MRI, just nicely introduced by Mario, we have a chance because we can actually push spatial resolution and hope to get to this. So the first project that we've been doing, and this is to start looking at the thalamic reticular nucleus. We have a number of atlases available, also post-mortem and MRI data that shows that you can nicely see the TRN, but we wanted to see if we can do this in vivo. So we've been collecting a lot of anatomical data. This is quantitative T1 data, a lot of repetitions. There's also quantitative T2 star data that's coming up in a second, and you can nicely see a lot of detail in subcortical structures there. This is 0.35 millimeter isotropic. This is work of Susanna, who is presenting the poster tomorrow, poster 60. She's back there. So please go see the poster if you would like to know more of this. But she actually took this data and tirelessly manually segmented the TRN, and you can see it here as a kind of a brighter structure. We have 11 data sets at the moment present, and she has segmented so far 5 of these. So I'll show you some of these results now. These are the segmentations projected on an MNI template. And overall, we see a lot of consistency in volume, in thickness as well, and also in shape. So you can see that actually if you look at coronal sections, you can see that the TRN more rostrally is really like wrapping around the thalamus, and as you go to the more caudal sides, it kind of arches out, wrapping around the LGN that you actually can see sitting there on both sides. It's a bit darker, and also actually curving around the MGB that sits just laterally of that. But you also can see here already that there are differences. So you can see that while the shape, for example, is very similar here across individuals, you can see like this little one sticking out. And also here you really see that the shape is kind of changing from this more round shape to this arching out shape at different rostrocaudal points across individuals. So there are differences. So first of all, I guess the consistency, it confirms the visibility that we can, feasibility of actually seeing this in individuals, but there is variability too. Susanna quantified this and can tell you more about this. But actually, if we look through Dice coefficient of the, which shows you the relative proportion of volumetric overlap, there you can see that overlap is not that huge. So we see about 20% overlap across individuals. However, if we quantify similarity with a different metric that not just counts the overlapping proportion of voxels, but actually captures alignment in shape, we see that the distance between individuals is really not that large in shape, and we're often under 1 millimeter between individuals. So the shape is the same, but they can shift a little bit in location or orientation relative to each other. And we believe that we're actually looking at true anatomical variation across individuals here. And that confirms that it's actually relevant to map the human TRN individually if we want to start exploring its function at some point. We started looking at function already using existing data, so looking at sound responses and seeing if we could see any overlap, and that is what is shown here. So to see, we do get sound responses in this TRN for some individuals, but we also are realizing that we actually, given the variability, we're going to need a lot of functional data in individuals for which we can actually segment the TRN. So we'll need to get data from the same individuals to make any clear claims on this. Of course, we would also like functional changes. And with functional MRI, we can also look subcortically at responses to sounds in MGB and inferior colliculus. Given time, I'm going to skip a little bit over this, over the paradigm, but feel free to approach me later in case you're interested. But we again set up an attention, is a frequency selective attention paradigm. This is work from Anna Maria that is also somewhere here, so feel free to approach her as well if you're interested. But now what we're looking at is not just cortex, but we're actually going down to check subcortical structures as well. Sorry, I'll have to click through this a little bit. Scanning a lot of data for each individual. So we have 5 sessions for each participant, where one session is just anatomy, and then we have about 6 hours worth of functional data, simply because if you go subcortical with small voxels, you need a huge amount of data to get any responses. And I think based on the first data that is coming in, we'll need even more than this at a spatial resolution of 0.8 millimeter isotropic. We see nice tonotopic responses, and inferior colliculus is here, MGB2, and based on that tonotopy, we can actually guess where ventral MGB is located in individuals. We also get cortex. Cortex always works, so you again see the tonotopic maps in one individual for baseline, attent low, and attent high. Interestingly, because we have now pushed spatial resolution so much, we can also do what was just mentioned already and go laminar with this on cortex. So we can actually get, sorry, I did not explain this, so I'll skip it. So we can do cortical depth-dependent analysis and actually start to see how frequency and population receptive fields change with attention in deep, middle, and subcortical and superficial cortical layers. So this is all very preliminary still, but first results are coming in, and I'm excited about it, so I wanted to show you what we're up to. So take-home messages. The effects of feature selective attention, I believe, strongly depend on stimulus and task. And in our hands, we saw a big difference, which we attribute to single versus multi-stream sound stimulation, resulting in either competitive enhancement or preparatory optimization. Ultra-high field MRI opens up the opportunity to look beyond auditory cortex in feature selective attention, and for me, beyond the auditory cortex will mostly be below auditory cortex, so that's what we're going to be trying for the next few years. Anatomical MRI for individual TRN segmentations, exploring the MGB and inferior colliculus for functional responses, and then linking this to cortical depth-dependent analysis in cortex, which should give us a handle on connectivity, right? So knowing how the cortex projects down and also receives input should give us interesting insights there. So thank you very much for your attention, and I'm looking already forward now to tell you a lot more about this at the next ICAC. Thank you.
We have time for a couple of questions.
Can I go? There's Maria has one. Is that okay? Ah, okay. Yeah, Maria has one already. Yep, go.
Thank you, Michelle. That was really interesting. I have a kind of cheeky question. So you're hypothesizing thalamus. Could it be, and I'm saying this completely in a completely uninformed, I'm not doing a lot of fMRI, but could it be that what you're seeing is prediction and so attention biases your prior to what you're expecting to hear. So for example, high frequencies. And so these reductions could be because you're minimizing prediction error. So then that would bias the hypothesis towards looking above auditory cortex rather than below.
Yeah, I think it could be. So it could be, I think, at this point, many effects. It would also mean that, though, that with that prediction, that the prediction would actually have the effect of sharpening, right? So that would actually be a very interesting thing too. So I think with starting to look at laminar effects and actually at this whole circuitry, we'll be able to get to that question, because we'll actually be able to see and specifically also looking at how connectivity changes with the different attentional conditions, we'll be able to get the circuitry out. And I think that will be informative on knowing what we're actually seeing. Absolutely, yeah.
Thank you.
This is really lovely. It's really exciting to see people pushing what you can do with fMRI in hearing. I had a question about your first conclusion about it being task differences and specifically single stream. You're doing this under continuous scanning, right? So there is, in fact, always a second screen going bing, 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 no matter how good your hearing protection is. And I wanted to hear your thoughts about that versus a presented, controlled stream.
You're absolutely right. We are doing this under the worst possible circumstances for auditory, for hearing, but so were all the previous functional MRI studies. So there is a difference, and I have a feeling, having been in the scanner many times myself. So one thing that we do on purpose is to actually set up these attentive frequency bands outside of the frequency of the scanner, right? Because if you don't do that, your signal kind of saturates there. And we have a feeling also that is what is happening, because if we look, I showed you before the sensitivity or the prediction accuracy that we actually have in our PRFs. And if you look at that in a frequency-specific manner, you always see kind of this drop for the frequencies that are at the scanner noise. So it has an effect. I have a feeling that by staying away from it, it helps. But yeah, if we could get a silent version, that would be awesome. Yeah.
Yeah, thanks very much for a great talk. I'm very interested in subcortical mechanisms, involvement in also speech and noise segregation. So I'm very interested in your thalamocortical circuit. Can you speculate, say, for speech-on-speech discrimination? Say you have different voices with different fundamental frequencies, different harmonic structures. Could the spectral resolution that you see in the MG beam and with the feedback, do you think that could play a role in speech-on-speech segregation when you have smaller differences, and if not, things like that?
Yeah, that would be, in the impact, right? And the grand proposal, so the impact of what this would ultimately have. So I think maybe we'll have to see, right? We're going to start with artificial sound, but the next step would really be to port this to natural sound settings, right? So to see if whatever mechanism that we see on these artificial amplitude modulated pure tones or filtered noise, if actually we can replicate that when we're using speech and competing speech in a healthy population, a healthy hearing population, and then it would be extremely interesting to see also, for example, how this changes with aging. And how this works. Yeah, that would be wonderful. I cannot speculate yet. I would like to say yes, but I have no data to pick that up.
I think you mentioned something with modeling. I'm not quite sure if I caught it correctly, but have people done modeling on these questions, or not so much yet?
Not that I'm aware of, because you would need a type of modeling, I guess, but I guess we can ask the audience. I'm sure that somebody knows a relevant reference. You would basically need to what you could do is basically change resolution of your or sharpness of your tuning and see for different sounds what the effect would be, right? And I guess the interesting thing would then be to see how quickly this resolution could change and how that would mean, how that would result in adaptation. I think you could do something. How accurate it then reflects biology, that would be how accurately it reflects biology.
Great. Thank you very much.
Last question back.
Hi, Michelle. Amazing as
Hi.
always. I was in your active attention condition. Do you see any changes in the voxels that prefer the unattended frequency? And I was wondering if, in particular, because you have, especially with your resolution, if you have something that maybe looks like inhibition of the unattended band, and if you would predict that would be in a particular layer, or, depth.
Yeah, great. That's exactly what we were kind of expecting to see, and we didn't see any of it. So what I showed now was not yet cortical depth-dependent fMRI, right? So these were voxels of the first study I showed were 1.2 millimeter isotropic resolution, so we couldn't do any cortical depth-dependent analysis there yet. But we did look for this, looked for gain effects across frequency, looked for shifts in frequency, not just at the attendant frequency, but also further out. And you do see some shifts at individual level, but nothing consistent across the participants. Yeah.
Okay, thank you. Please